Hi, this is Mr. Worski again. This is the second in the photosynthesis series of screencasts. Last time we were here, we were almost finished with the light-dependent reaction. We covered everything from photosystem 2 here, which actually happens first as P680, to photosystem 1. They think that is the first one that evolved. That's why they call it photosystem 1. And we covered from here in the formation of NADPH. So the last thing we need to talk about in the light reaction is photosynthetic phosphorylation. And what we're going to do here is we are going to produce ATP with the buildup of the hydrogen ions in the inner space. So we know that there is a buildup of hydrogen ions in the inner space. We know what steps cause the buildup of hydrogen ion in the inner space. We have the movement of the hydrogen ions from the stroma to the inner space by PQ, as well as the breaking up of water to supply P680 with electrons. Now, since there is an increase of hydrogen ions in the inner space, we can actually measure the pH, because remember, at high levels of hydrogen ions, we have a drop in pH. And in fact, the inner space pH is a pH of 5, while the stroma has a pH of 8. So there is a lot of potential energy here because the hydrogen ions want to move out, but they can't move through the thylakoid membrane because they are polar, and they won't be able to deal with the hydrophobic middle layer, which is hydrophobic. So they need a tunnel, right? So that would be facilitated diffusion. Good. The tunnel that they use is called ATP synthase. Now that name implies that ATP is synthesized, and that's true. So ATP synthase looks like here we have a rotor, which is in the membrane. We have the top here, which is the knob. And then we have the rod in between here. Now this is going to provide a tunnel. The stator here keeps it in place. Now this is another example of a wheel, because as the hydrogen ions move through, I don't know if you can see this here, moves through here, that it's going to rotate. And as it rotates, it's going to physically shove a phosphate onto an ADP to form ATP right here. So as the hydrogen ions move through, the ATP synthase rotates, and as it rotates, it will physically jam the phosphate on top of ADP, which is adenosine diphosphate, to form ATP, which is adenosine triphosphate. It moves in a clockwise fashion, and this is another example of a wheel in nature. So you have two. Somebody asks you, do wheels naturally happen? You say yes, prokaryotic flagella as well as the ATP synthase. So this is the overall look at the light reaction, in which we take light, we take water, NADP, plus hydrogen ions, plus ADP and phosphate to get ATP, NADPH, and oxygen. Okay. So now the NADPH and ATP move to the dark reaction, or the dark side, and oxygen will go to the atmosphere and allow us to breathe and use it in the production of energy. And here we know exactly what happens now. We should be able to explain all of this stuff in this diagram. That's what we learned. Take a look at that, draw it, do something with it, but you should be able to explain everything. The dark reaction, or the light independent reaction, will take the ATP and the NADPH. We're going to add carbon dioxide, and you're going to produce glucose. There are two types of dark reactions. There's C3 and C4. C3 happens in every single organism that does photosynthesis. So we're going to focus our attention on C3, which is called the Calvin cycle, and it is named after a 3-carbon compound called phosphoglycerate. C4 is named after a 4-carbon compound called oxaloacetate. But we're going to first talk about C3. In C3, or the Calvin cycle, we're going to start off with a 5-carbon sugar called ribulose biphosphate, or RUBP. We'll add carbon dioxide from the atmosphere to RuBP, and you're going to form an unstable 6-carbon compound that immediately breaks down into two 3-carbon phosphoglycerates. The enzyme that adds the carbon dioxide to RuBP is called RuBP carboxylase. It has a nickname which is called Rubisco, which is the most abundant enzyme on this planet. That's a fun fact. We will take 2 molecules of 3-phosphoglycerate. The reason we call it 3-phosphoglycerate because the phosphate is on the 3rd carbon. And we're going to add 2 molecules of ATP. We're going to remove a phosphate, put it on the 1st carbon, and we're going to form 2 molecules of 1,3-diphosphoglycerate, which means that the phosphate is on the 1st and 3rd carbon of this 3-carbon molecule. And 2 ADP. Now the 2 ADP will go back to ATP synthase so that we can have another phosphate shoved on top so that they can form ATP again. The 2 molecules of 1,3-diphosphoglycerate will have electrons added to it, 2 electrons, and the electrons come from NADPH. You will form 2 molecules of phosphoglyceraldehyde. 2 phosphates are lost. Those phosphates go back to AP synthase so they can be added to the ADP. 2 electrons are given to the phosphoglycerate, and the hydrogen ion goes back into the stroma so it can be moved back through the thylakoid membrane, or it can be added to NADP. And you're going to form also 2 molecules of NADP+, which go back to accept the 2 electrons and the hydrogen ion from ferredoxin. Whew. That's what we have so far. We're up to this stage right here. And this is all the stuff that goes on. But we're not done yet. We're almost done, though. Now, we form phosphoglyceraldehyde. This happens 6 times to give us 12 molecules of phosphoglyceraldehyde. Now remember, each phosphoglyceraldehyde has 3 carbons, right? So 10 of those phosphoglyceraldehydes, which means 30 carbons, goes back to form RuBP. Now remember, RuBP has 5 carbons each, so 5 times 6 is 30, so the number of carbons is conserved. That gives us 2 phosphoglyceraldehydes left over, and those 2 phosphoglyceraldehydes will go form a molecule of glucose. And there's the equation down there. 6 RuBPs plus 6 CO2s yields 12 PGs plus 12 ATP, we get 12 DPGAs plus 12 NADPH yields 12 PGAL and 12 phosphate. So now you should be able to tie both the light and dark reaction in together. And the light reaction produces NADPH and ATP, whereas the dark reaction takes the carbon dioxide, the ATP, and the NADPH to form glucose. Now, the C4 pathway is another step added to the C3 pathway. It's a more efficient way of moving the carbon dioxide from the leaf into the cell. So just add 4 extra steps. So we have CO2 plus PEP, which is phosphoenolpyruvate. We form a 4-carbon oxaloacetate. The enzyme is PEP carboxylase. The oxaloacetate is added to NADP, and the oxaloacetate gives up 2 electrons and a hydrogen ion to NADP to form malic acid, or malate. The malic acid will give up the CO2, and that CO2 will go to the C3 cycle, and that forms pyruvate. Pyruvate is added to ATP to form phosphoenolpyruvate, which goes right back up to step 1. So you might be asking yourself, well, where does this, what's going on here? And that's a really good question. So we need to know about a leaf cross section, and I think I have one on the next, sort of. So the CO2 enters the leaves through these holes at the bottom of the leaf called stomata. The carbon dioxide combines with the PEP in the mesophyll cells. Malic acid is stored in bundle sheath. CO2 enters the mesophyll to join with RuBP carboxylase. Why did we do this? Or why do leaves do this? Why? Because it removes the CO2 right away when it enters the leaf. And if it removes the CO2 right away, it decreases the carbon dioxide level inside the leaf, so it increases the rate of diffusion. That's why leaves do this C4, if they're C4 plants. Only C4 plants can handle it. I mean, C4 plants are ones that can handle hot environments. So we have the bundle sheath here and the mesophyll here, and the CO2 is removed right away from the stoma, or the leaf space here, which decreases the carbon dioxide level, and then it shoves the CO2 directly to the C3 cycle, which then forms glucose. So the C4 plants have an extra 4 steps in addition to the C3 steps. All organisms that photosynthesize do C3, only some, the C4 plants, do the C4 cycle too. So photorespiration, there's a big mess up here. And RUBP binds with oxygen instead of carbon dioxide, forms glycolic acid. It has to be changed back to RUBP before it can go through and form glucose. And this just decreases the efficiency. So in hot environments, it is better to remove the CO2 right away and then decrease the concentration of CO2 so that increases the rate of diffusion. So crabgrass is a C4 plant. It is perfectly adapted to hot, dry environments, and they thrive in those environments because it's a C4 plant. And that is the end of photosynthesis. I know, sad, very sad. If you have questions, please refer to your textbook. You can ask me tomorrow in class or watch the video over again and listen to my voice. I'll see you tomorrow. I hope this